Alright, let's finish up our last major chapter, chapter 17, with concept 17.5. It's all about the things that can go wrong. This transcription translation process is very complex, and it's got a lot of different steps, which means there's a lot of possibility for problems. So we're going to talk about a few of those problems. It says 17.5, mutations of one or a few nucleotides can affect protein structure and function. And that makes it sound not so bad. Protein structure and function can be affected. Yeah, well, that can either be fatal or it can result in horrible problems, issues, diseases, or various symptoms with the living creature there. Oftentimes, if this occurs in the developing embryo, it results in a spontaneous abortion or death of the embryo. And then if it occurs, if it's livable or it's not such a change that the embryo is spontaneously aborted, it can result in some pretty bad physical and mental manifestations. So let's take a look at what they're talking about here. Number one, mutations are changes in the genetic material of a cell or a virus. We should be familiar with that phrase already. We're going to talk about a couple of different types of mutations, and some of these only affect one base pair or one tiny section of the DNA. Remember, a base pair is like right here. So this portion right here with A connected to T is a base pair, or G connected to C is a base pair. So you can have one tiny problem right there in that one base pair, and that can cause some pretty dramatic effects. Or you can have larger mutations that affect larger pieces of the DNA, and we'll talk about both. The first one is a point mutation. Point mutations are chemical changes in just one base pair. So a point mutation is the smallest kind of mutation you can have. It's a change in one of these base pairs of the DNA. The change of a single nucleotide in a DNA template can lead to the production of abnormal proteins. Remember, this is the instruction code that is going to be then created into messenger RNA and then sent out to the protein manufacturing units, the ribosomes. And if that instruction code is off by even one letter, it can create quite a bit of problem. If you recall back in chapter 15, we were discussing chromosome mutations, and that would be affecting generally large sections of DNA, long segments of DNA. We would think of those as large-scale mutations. So here we're primarily talking about small-scale mutations, but that doesn't mean that they can't be just as devastating. Let me give you a single example of a disease or a issue that we've already talked about, sickle cell anemia. Sickle cell anemia we've discussed is a problem where the hemoglobin of the red blood cell is misshapen, and it causes the red blood cell to be kind of crescent shaped. It can't carry oxygen as efficiently, and it tends to clog in capillaries and cause pain and other symptoms. And we've discussed how that can be a very bad condition, and it's a genetic condition that can be passed on. Well, that entire condition and that problem is a point mutation. It is caused by a change in one single base pair. So if we take a look at the DNA of the wild type, which would be what we would consider the normal type of hemoglobin in this particular area, it's CTT, which corresponds with GAA down on this strand. Well, the messenger RNA that's going to be created from that, this top strand is our template in this case, would be GAA. And GAA codes for this particular amino acid, glutamine, I believe is what it is. So that glutamine in that spot gives you normal hemoglobin and everything is fine. Now take a look at this DNA strand right here. Instead of having the T and A down here, they are swapped. And you would think, well, it's still the same base pairing, A with T, but they are in the opposite position of where they should be. How does that affect it? Well, remember the top strand here is our template. So this is the mutant hemoglobin DNA. The top strand is our template to make our messenger RNA, and that's going to code for GUA. Now remember, the messenger RNA doesn't use T, it uses U instead, so GUA. Well, GUA codes for this particular amino acid, VAL, and I'm not even going to try to recall what it is. I'd have to look it up. There's 20 of them, so it's hard to remember all of them. But what happens is, with that amino acid in place, that causes sickle cell hemoglobin. So that one change in that one amino acid caused by the one point mutation in the DNA causes an entire disease that can be pretty debilitating if you have this disease. So remember, we talked about the genetics of this. For this disease to show, for you to have the full spectrum of the disease, you'd have to be homozygous for this trait. So both your mom and dad would have to have the mutant hemoglobin DNA, and that got passed down to you, and you would have the sickle cell disease. But it's amazing that that one tiny point mutation can cause such a major problem. This point mutation, this small scale mutation, there's basically two varieties, two ways that this can go wrong. And so we're going to look at the two categories. The first one is what we call a substitution, a nucleotide pair substitution. And then the other one is called an insertion or deletion. So we're going to look at one variety called substitutions, and then we're going to look at another variety which would either be insertions or deletions. You can kind of tell from the names what this means, but we'll go into a bit more detail. So let's talk about the substitutions first. A nucleotide pair substitution replaces one nucleotide and its partner, its matching partner, with another pair of nucleotides. And that would be similar to what we just saw with the sickle cell anemia. So this is a substitution error right here. We have what should be TA substituted with AT. It still codes for an amino acid, but it codes for the wrong amino acid, and that causes a problem in our hemoglobin of the red blood cells. There's actually three possibilities when you have a substitution. You could have what we call a silent mutation, where it doesn't have any effect at all. Remember, there's a lot of redundancy in the genetic code. We have a number of different 3 codon segments that code for any one particular amino acid. And so oftentimes mutations don't hurt you or help you. We would call these silent mutations, and they have no effect on the amino acid because of some of the redundancy built into the genetic code. The other type of mutation is called a missense mutation. This would be where it codes for an amino acid but not the correct amino acid. And that would be like the sickle cell anemia substitution. That's a missense. It's still coding for an amino acid, but unfortunately it's not the right amino acid. So that example of the one we just looked at with the sickle cell anemia is a missense mutation. And then the third mutation is a nonsense mutation, where it changes the amino acid codon maybe into a stop codon or some other problem, and that's almost always going to lead to a non-functional protein. So if you have a 3 codon code that's supposed to code for a protein, and the mutation causes it to be one of the 3 stop codons, well, then the whole process is going to end, and the ribosome is going to get to that point and stop making the polypeptide chain, and then you're not going to have a complete protein. So a nonsense mutation is probably going to cause a complete collapse of the creation of that protein, and it would be a non-functional, non-working protein. So let's take a look at some of those substitution examples. The first one is a silent mutation. It doesn't have any effect at all. So for example, here's our mutation right here. Here's the wild type. We're going to be looking right up here. And this is the mRNA that gets created. And then this is the proper amino acid chain, the polypeptide chain that gets created. So let's look down at the substitution error that occurs. So right here, we have a substitution where we have an A instead of a G. Well, what that means is when we create our mRNA, it doesn't really have any effect because, remember, that would normally be a T, but because the mRNA doesn't have the T anyway, it's going to put the uracil there. So the uracil would be there no matter what, so it doesn't have any effect on it, and it codes the same way. So you get the same amino acid chain, you get the same polypeptide chain, and that mutation didn't have any effect, and you wouldn't even know it by looking at the, say, the phenotypical process of creating the protein. You wouldn't even know. But if you check the actual DNA, you would see the mutation, but it's a silent mutation that has no outward effect. The next substitution problem would cause an amino acid to be created, but the wrong amino acid. So here at the top we have the wild type, the normal version, and you can see the chain of amino acids that gets created, the polypeptide chain in the protein. And then down at the bottom, here is our substitution. We should have a C here, but instead we have a T. So that codes for an A down here in the messenger RNA. And that 3 codon triplet there codes for this particular amino acid. So notice they all match except for the last one. So it's creating an amino acid chain, but it's inserting the wrong amino acid in this particular case, and that could cause problems. So this is a substitution that results in missense. The last type of small-scale substitution mutation results in what we call nonsense. And in this case, here is the wild type, what we should be coding for. Here is our error. Right here we have an A instead of a T. So then when the messenger RNA codes with that A, it puts in a U right there. Well, the problem is UAG is a stop codon. So the AUG is correct, and it puts the methionine in there like it should. But then it gets to UAG, and the ribosome reads that as a stop codon, and so it stops. And so that's it. You have nothing. You have no functional protein because it got to this point, and it read UAG, which is a stop codon, so it didn't continue. So this mutation causes nonsense and a non-functioning protein. Those were substitution errors, and those can be pretty bad, but sometimes they have no effect at all. Let's get to the more serious errors, which would involve insertions and deletions. Insertions and deletions are either additions or losses of nucleotide pairs in a gene, and these, generally speaking, have disastrous effects on the resulting proteins. This is almost always bad news. Insertion or deletion of nucleotides can alter the reading frame. Remember, we talked about how you have to read these triplet codons in the right way. Just like you have to read a string of letters, they have to be put together in the right way to form words, to form sense, to pass on information. Well, if you have an insertion or a deletion, you produce what's called a frame shift mutation, and this is generally bad news. A frame shift mutation is basically shifting the reading pattern, and so you get gobbledygook instead of useful information. So a way to think about this is imagine shifting the reading of the following sentence one letter to the right. So I chose a sentence that has 3-letter words, just like the triplet codons of the messenger RNA. And so I chose this sentence: the cat has fur. Now, if you start with the T and you know every 3 letters is a word, well, then you can read this: the cat has fur. Even if I ran them all together, you would know that every 3 letters is a new word, and you could read it as the cat has fur. Or the cat ate mud. So I could even put 2 sentences together. I could run all of these letters together, and as long as you know you start with this T, and every 3rd letter starts a new word, you could read these 2 sentences: the cat has fur, the cat ate mud. But what if we do a frame shift mutation? What if we shift to the right by 1 letter? We follow the same rules. Every 3 letters we start a new word, and we're reading from the right to the left, but all we did was shift over 1 letter to the right. Well, then we get this: heck, ash, heck, ata. We get nothing. We get gobbledygook. That's a frame shift mutation. So just like these are 3-letter words, and even if I took out all of the spaces, just like this, we could read these 2 sentences if we knew to start with the T and that every 3 letters was a new word. The cat has fur, the cat ate mud. But if we shift by 1 letter to the right, suddenly it makes no sense at all. That's what happens when you have an insertion or a deletion. In this case, it would represent like a deletion. And so now we have to follow the same rules of reading the sentence. We get no useful information. Here's some quick examples similar to looking at the other examples. Here's our wild type, the proper, what we want to see with the letter formation and then the amino acids creating the protein. Here we have an extra A in place, so we have an insertion. Well, then that's going to code in this spot for a U, and we have a frame shift, and then we have a stop codon. UAA is a stop codon, so immediately we get nothing. We have a stop right there, and we have a non-functional protein. We have an immediate nonsense right off the bat. Here we have another example where we have a missing particular letter. We have a missing base right here. There should be an A, so that's missing, and so that codes for a U that should be there, but it's not there. So we still have a frame shift, and this happens to code for amino acids, but they're not the proper amino acids. So we get extensive missense. In other words, once we get to this point, It may code for amino acids, but suddenly they're all the wrong amino acids because we were missing a letter. Here we have an example of a whole section missing. We have three base pairs missing here, TTC. So we're going to have a missing AAG in your messenger RNA. This creates a frame shift, but one amino acid is, no frame shift, but one amino acid is missing. So we didn't shift our reading frame, we just dropped a whole word. So in this case, this would be like our sentence, and these are our words. Well, we have the proper words, but we're just missing one whole word. So no frame shift, but just an entire amino acid missing. So here are all those examples. You can kind of take a look at them together. I kind of look at them in the bigger one where you can see it better, but here's a summary of some of those examples. So we have substitutions, which tend to be pretty bad, but not as bad as insertions or deletions, which tend to be fairly disastrous because they cause oftentimes frame shift mutations, which end up causing a world of problems. Finally, regarding mutations, just a quick note about mutagens. Spontaneous mutations do occur during the DNA process. It's a complex process, so occasionally you're going to have mistakes that occur naturally between replication and recombination. You remember back in Chapter 16 we talked about the cell has a variety of repair mechanisms to try to keep these natural mutations down from happening, but some of them do get through and persist, and if these mutations occur in the germ cells that go on to create either sperm or eggs, and they can be passed on to future generations. Then we also have environmental factors called mutagens that can cause this to happen. These are physical or chemical agents that can cause mutations, various forms of radiation, everything from UV radiation from the sun to other forms of radiation that are present in our natural world, various chemicals, things that cause cancer oftentimes tend to be mutagens. A lot of mutagens are carcinogenic. So, for example, smoking produces carcinogenic chemicals that can get into your lungs and cause mutations and cancers and the like. So, some of these mutations are naturally occurring, and some of them are caused by environmental factors called mutagens. We're going to go ahead and finish up. There was one more portion here, Concept 17.6, and we'll go ahead and add it to the end of this one because we're almost done. It says, while gene expression differs among the domains of life, the concept of a gene is universal. So if we think about the three domains of life, that would be eukarya, archaea, and bacteria. I think I even have a website pulled up here. Let's take a look. So eukaryotes, bacteria, archaea, the three domains of life. Obviously, there's some differences between these three domains. Gene expression occurs slightly different amongst these three domains, but the concept of a gene, a segment of DNA which gets transcribed into messenger RNA and translated into proteins, that is consistent and universal across all life. Archaea are prokaryotic creatures, but they kind of actually fall somewhere in between the gene expression features of a eukaryote and a prokaryotic creature. So many of their features seem to fall in between, and there are differences between these three domains of life, but this idea of kind of this central dogma idea of DNA to RNA to protein, you can find in pretty much any living creature on Earth. We know a lot more about the gene expression of bacteria and eukaryotes than we do the archaea, but we have done some study of the archaea, and we can make a few assumptions based on what we see. Bacteria and eukarya differ in their RNA polymerases, so some of the various enzymes kind of differ. They differ in the way they terminate transcription. Remember, the bacteria, when transcription is done, basically that messenger RNA is ready to go. But in eukaryotes, there's a lot of transformation of the messenger RNA. You have the end caps that are added, and you have the various introns that are cut out and things like that. And some of the ribosomes are a bit different between the bacteria and the eukarya. The archaea tend to resemble the eukarya in these respects. So the archaea seem actually to have more in common with the eukaryotic creatures than the bacteria, but they tend to sort of fall in the middle sometimes. Remember, the bacteria can transcribe and translate at the same time, the same gene, because there's no nucleus. So this process can actually occur at the same time. Whereas in human cells and other eukaryotic organisms, transcription and translation happen in two different places because we have a nucleus, we have a nuclear envelope. transcription happens inside the nucleus, and then the mRNA heads out to the cytosol material and does translation. In archaea, we don't know as much about the process, but it seems like transcription and translation are likely kind of coupled together, more like in the bacteria. So even though these three domains of life differ in some respects, a lot of the process is the same. And again, that goes back to this idea of evolutionary theory and the idea of common ancestry, and how so much of our genetic code and the genetic code of creatures that are as different from us as an oak tree or a fruit fly, we still share a lot in common genetically with those particular creatures. Another great example of an actual photograph taken from a microscope and an illustration, this would be in bacteria, where we have the DNA strand, that very thin strand you can see up there, and we have RNA polymerase creating the messenger RNA. So we have transcription going on, and look, there's ribosomes attached to that messenger RNA as it's being created. So they're doing translation. So it's kind of like they're both happening at once. As that RNA polymerase moves along the DNA and creates the messenger RNA, the ribosomes are already on that messenger RNA spitting out those polypeptide chains. So we have transcription and translation happening all at the same time, all in the same place, and you get to see it through the microscope right here. Of course, we know in our cells, in the eukaryotes, it's different. We have transcription happen in the nucleus, and then we move out to the cytoplasm for translation. So let's finish up with one last question. What is a gene? Well, that's a good question. The definition depends on how exactly you're looking at it. The idea of a gene has changed over time. One of the strengths of science is that our information changes. Some people consider that to be a weakness, and they say, well, science can't be trusted, or you can't rely on it because in 20 years the information will be different. But that's a totally wrong way of thinking about science and the scientific method. That is actually the strength of science, is that we continually update and add and modify what we know to better reflect reality. So the definition of a gene has changed over time. And you could consider the definition of a gene to be different things depending on how you were studying or what you were looking at. So we have considered a gene as a discrete unit of inheritance when we were discussing Gregor Mendel, and that's still a good definition. We have considered a gene a region of a specific nucleotide sequence in a chromosome. When we were talking about the chromosomes and what that meant, and we were studying chromosomes, that's a good definition for what a gene is. And then finally, now we're talking about a gene as a DNA sequence that codes for a specific polypeptide chain. That, too, is a proper definition, probably the most accurate modern definition. But all of these are good and helpful definitions depending on the context in which we're studying the genes. So we're going to end right here with another great kind of summary of transcription and translation in the eukaryotic cell. It's amazing to look at this. You can see this on your text, page 348. And if you haven't studied this chapter, just to turn to this page and look at this illustration, you would think, okay, there's no way I can understand this. But if you look at it, you can look at the individual parts of transcription and then translation, and this is something you can actually understand. So after reading through this chapter and watching the videos, you can look at this and you can say, yeah, I think I kind of understand what's going on. Well, you should pat yourself on the back and feel good about yourself because you're making a lot of progress, and I'm really proud of you. I'm really proud of how hard you guys have worked all this year. I know it's been a tough course, and taking it online has made it extra hard, but I hope what I've done with these videos and with everything I've done online has been helpful, and I'm looking forward to the second semester, and I'm looking forward to a few changes I'd like to make to hopefully make the process smoother for everyone and all of us, and we'll get ready to go. We're going to look at Chapter 18 really briefly, and I'll tell you what's coming up with that. I won't have many questions about Chapter 18 on the final exam, but we do need to hit some of the highlights. Our last slide right here, again, talks about the central dogma that we have discussed, DNA to RNA to protein, and in summary, a gene can be defined as, here's a great ending definition, a region of DNA that can be expressed to produce a final functional product, which will either be a polypeptide or an RNA molecule. Do yourself a big favor and me a big favor. Check out the crash course and the Bozeman Science videos about transcription and translation. They are really helpful in clarifying this information and the process involved. Like I always say, pretty pictures and funny words and clever animations are always very helpful. So one last thing we're going to take a look at in Chapter 18 is a little bit about how do we control the expression of these genes. So think about every cell in your body has the full genetic code, right? So your skin cells, your eye cells, your fingernail cells, your bone cells, they have all the instructions for creating your entire body. A given type of cell only expresses a small subset of its genes. So they have the entire code, but they don't use the entire code. Imagine the problem you would have if the lens cells in your eye began to express the genes for hair proteins, which are usually only expressed in the hair follicle cells. If suddenly hair started growing out of the lens of your eye, you would have a problem. Well, the proteins, the instructions for making the proteins for creating hair are in the lens cells of your eye. So what keeps that from happening? What controls gene expression? Well, we're going to find out that gene expression is very tightly and precisely controlled and regulated. That's what Chapter 18 is about. We're only going to hit the bare highlights, and then we're going to end our semester. So again, I appreciate all your time this semester. Good luck on your exam. I'll have that to Ms. Restivo very soon. Study, study, study. I will be online Sunday night, and we have one final quiz that ends Sunday night as well. I hope to talk to you online or via email very soon. Bye-bye.